The overall goal of CARIP-Seq is to survey genome-wide chromatin-associated RNAs. The goal of ChIP-Seq is to evaluate protein-DNA interactions on a global scale in embryonic stem cells.
These methods can help answer key questions in the epigenetics and stem cell fields, identify the genome-wide location of transcription factors, or epigenetic regulator binding, or histone modification occupancy, or chromatin-associated RNAs in embryonic stem cells. The main advantage of these techniques is that they allow for the genome-wide, unbiased interrogation of RNAs associated with chromatin constituents and global identification of protein-DNA interactions. Though these methods can provide insight into embryonic stem cell biology, they can also be applied to other mammalian cell types to study cancer biology or other human diseases.
To harvest the embryonic stem cells, wash the cells first with phosphate-buffered saline. Then, detach the cells with 0.25% trypsin, pre-warmed to 37 degrees Celsius. After trypsinizing, resuspend the cells in pre-warmed MEF medium supplemented with 10% fetal bovine serum. Then, add 37% formaldehyde to the cells in a conical tube to a final concentration of 1%. And incubate at 37 degrees Celsius for 8 minutes to cross-link the cells. To stop the fixation reaction, add 1.25 molar glycine pre-chilled at 4 degrees Celsius to a final concentration of 0.125 molar. Then, place the tube on a rotator and invert it at 8 revolutions per minute for 5 minutes.